In the previous video, we saw what happens at the microscopic and nanoscopic scales when animals eat food. We fed potatoes to mealworms, and we used a scale and BTB to determine what comes in and out of the mealworms. So what did we learn from that? Let's think in terms of the mealworm's cells. We found that oxygen and sugar enter the cells, and we saw also that carbon dioxide and water came out of the cells. We determined that sugar brought in food energy, and that energy was converted into useful heat and movement for the cell. But there was one more very important thing that we observed, and that is that the mealworms gained weight through the course of our experiment, and it is certainly worth our time to explore why. Before we go any further, let's identify the goal for this video. After watching this video, you should be able to use a molecular model to explain how animals grow. Since we're talking about molecular models here, we'll spend quite a bit of time at the nanoscopic scale, which means that we'll need to use our molecular modeling kit. We can achieve our goal by appealing to some basic principles of chemistry. Here's one we've seen before.
Matter can be created or destroyed.
The mass that the mealworms gained could not have come from nowhere, so where did it come from? This is where chemistry comes in handy. In our previous video on molecular models for animal respiration, we saw a long-chain molecule called starch. It makes up most of the mass of potatoes, the worm's favorite food. And starch is really just a chain of sugars. The starch, being made of many units, we call a polymer. Polymer is Latin for many parts. The individual sugar units that starch is made of, we call these monomers. That's Latin for one part. Different chains of monomers result in different polymers. There's a wide variety of monomers and polymers, but in this video we'll mostly stick to carbohydrates, which is what starches and sugars are. Now it turns out that all foods are made of polymers, and all animals are also made of polymers. So how do food polymers turn into animal polymers after the animal eats them? Take a moment to pause the video and think about this question. One thing we need to address is the fact that food polymers turn into different polymers inside the animal after they're eaten. This requires breaking food polymers down into monomers. And then those monomers need to be rebuilt into polymers which are useful for the cells of the animal. Here's a helpful metaphor. Suppose you had a toy you made out of some building blocks. You'd like to build a new toy, but first you have to take this one apart. Taking the first toy apart is a bit like breaking down a food polymer into monomers. Building the new toy is a bit like the animal's cells using the monomers to build a new and useful polymer inside the animal. So now we have a brand new polymer, even though it's just made up of the pieces of the original food polymer. So how do animal cells do this? How do they take polymers and break them down into monomers and then build them back into new polymers? One of these processes you've probably already heard of. It's called digestion. In this process, the mealworm takes food polymers, starches in this case, into its stomach by eating them. Chemicals in the stomach called enzymes use water molecules to break down the polymer into the monomers it's made of, in this case sugars. Once the polymer has been broken down into monomers, these small pieces are absorbed into the bloodstream, where they can now make their way to the animal's cells. When these monomers enter the cells of the animal, there are several things that can happen to them, depending on what the cell needs. In the previous video, we already saw one very important way in which these monomers can be used. In the process we learned about, sugar combines with oxygen, and the sugar's energy is used for movement and heat. Carbon dioxide and water are waste products, which leave the animal. This is how your cells, and in fact all cells on Earth, get energy. And this process is called cellular respiration. Another thing that monomers can do is actually change into different monomers. You might have heard that eating a lot of sugar or drinking sugary drinks can cause you to gain weight in the form of fat. But how can this make sense when sugar and fat are completely different monomers? The reason for this is that your cells take in the sugar molecules and change them into fat molecules for future use. The fat molecules are stored in fat tissue, and the result is that you pack on some weight. Now, if you are trying to lose weight, you can convert the fat in your cells back into sugars, which are then used for cellular respiration. You can usually accomplish this with lots of exercise. But what about when animals grow in size, as in if you're trying to build some muscle? In this situation, monomers are bonded together to form brand new polymers. The general term for this process is called synthesis, or putting together. But since it occurs in living cells, we call it biosynthesis. So before we go any further, let's stop and consider all of the pathways that a polymer can travel as it is processed in an animal's cells. First, the polymer has to be digested into monomers. Most of the monomers will probably be used for respiration, since that's the single most important process for providing energy for cells. Some of the monomers will also be used to make new polymers in the process of biosynthesis. Finally, any monomers left over after respiration and biosynthesis will be changed to different monomers and stored for later use, sometimes as fat. And if you're trying to lose some weight, you can reverse that trend with heavy exercise. So if we label all the major pathways that food molecules can travel through the molecular machinery of the cell, it might look a little like this. Now that we know the basics, let's look at the processes of digestion and biosynthesis in a bit more detail. If we're really serious about understanding these molecules, we can actually build them with our molecular modeling kit. Digestion takes place in the stomach, so let's start there. Remember that food enters the stomach as a polymer, but this polymer has to be broken down into monomers to be of any use to the animal's cells. Well, how does this happen? To split a polymer into monomers, what we really need to do is break bonds. And in a food molecule, digestion is able to break those bonds specifically which link the monomers together. And in this particular molecule, starch, those bonds are located here and here. To digest this food molecule, something needs to cut these bonds right here. Biological chemicals called enzymes do this. Enzymes often act like a pair of molecular scissors cutting very specific bonds. It's a common misunderstanding that enzymes are somehow alive, but they aren't. They're really just chemically active proteins found in living things. The enzymes break the bonds in the polymer, and so the monomers are separated. At this point, we are left with only monomers. In other words, the starch has been digested into sugars, and this is because the enzymes broke the bonds between the monomers. At this point, we now have a problem. According to our rules of molecular bonding, certain atoms are required to have certain numbers of bonds with other atoms. Hydrogen has 1 bond, oxygen has 2 bonds, and carbon has 4 bonds. And in this molecular modeling kit, hydrogen is white, oxygen is red, and carbon is black. With this in mind, do you see any atoms in the new monomers that violate the rules of molecular bonding? Pause the video and see if you can spot the atoms that don't have the right number of atoms bonded to them. If you count the number of bonds, you'll notice 4 separate atoms that don't have the right number of bonds anymore. This oxygen and this oxygen each only have 1 atomic bond, but each oxygen atom is supposed to have 2 bonds. This carbon and this carbon each only have 3 atomic bonds, but each carbon atom is supposed to have 4 bonds. To resolve this problem, what we really need is more atoms. Fortunately, there are other atoms inside the stomach where digestion is taking place. These are found in water molecules, H2O. The oxygen and hydrogen in the water molecules are atoms that can fill the bonding gaps in our monomers. The enzymes break some of the bonds that hold the water molecules together. The resulting unbonded atoms can now squeeze into the incomplete monomers, forming new bonds. Now, every atom in our monomers has the right number of bonds. Digestion is sometimes called hydrolysis, and that's because water is involved in splitting up the polymer. We can even write out digestion as a chemical reaction. We start with a polymer, and we really add water to it. In the process, the polymer is split into monomers. So that's how we can write out digestion in the form of a reaction. That summarizes how the polymers in food are changed into monomers that the animal can now absorb and use. Let's get to the growing part. Let's keep talking about the nanoscopic scale, but move out of the mealworm's stomach and into its cells. After digestion, the monomers travel through the bloodstream into the cells of the animal. The most important process that the cell performs is cellular respiration for energy, but any molecules left over are used to build bigger molecules in the process of biosynthesis. Biosynthesis is necessary for doing things like building muscle. Let's look at biosynthesis in more detail. Biosynthesis is pretty much the opposite of digestion. Instead of splitting a polymer into smaller monomers, the cell puts monomers together to create new and useful polymers. Polymers are building blocks that the cell uses to create and repair things, such as organelles and membranes. Now imagine that the cell has 3 sugars floating around inside, but it needs to make a polymer out of them. Here's how the cell does it. Remember how with digestion there were special enzymes that split bonds? Inside cells, there are different enzymes whose job is to make new bonds. The same bonds that the digesting enzymes broke earlier are reformed by the enzymes in the cell. Now, through biosynthesis, the cell has started to create a new and useful polymer that it can use to grow. Let's take note of a few details about the process of biosynthesis. The most important thing to note is that a new bond was formed here, between the 2 monomers. They have now been joined into a simple polymer. Another thing to note is that in this case, a byproduct was also created. See how the atoms that were broken off formed H2O here? This means that any time biosynthesis takes place to bond polymers, water will also be created as a byproduct. Watch carefully as the enzymes bond the last monomer to create the new polymer molecule. This is the completed polymer, and now the cell can use this polymer to build new membranes, fibers, organelles, enzymes, all the parts it needs to function and do its job. Like digestion, we can also write out biosynthesis as a chemical reaction. In biosynthesis, the cell combines monomers together to produce a polymer, while waters are also made as byproducts. And if you look closely, biosynthesis is just the chemical reverse of digestion. Their reactants and products are opposites. It's no wonder we observed that the mealworms increased in mass. As they ate and digested the food, their cells built lots of polymers, and the mealworms grew in mass. We were able to detect this change in mass with our scale. We have one last thing to learn today. Let's take a look at the different types of polymers that might be made in biosynthesis. All of the following polymers and monomers can be found in food. One of the most important polymers that cells build is called protein. Protein polymers are built from monomers called amino acids. This is what amino acids look like. The blue atoms are nitrogen. And this is what the protein polymer looks like. Notice that it's just a chain of amino acids. Another polymer that cells can build is fat. Fat is synthesized by putting fatty acids together. Notice that fat contains all the same atoms as sugar: carbon, oxygen, and hydrogen. That's why cells are able to convert sugar to fat and vice versa, depending on what the cell needs. A fat polymer looks like this. Usually a fat polymer is made up of 3 fatty acids. As such, we call these polymers triglycerides. This next polymer you've probably heard of: DNA and its close cousin, RNA. These are the chemicals that store genetic information in cells. These important polymers are built from monomers called nucleic acids, because these monomers were first discovered inside the nuclei of cells. This is a computer rendering of a DNA molecule. The last polymer we've already seen. Starches are carbohydrate polymers most abundantly found in plants. Sugar monomers are synthesized into starch polymers. This is what our sugar monomer looked like, and this is what our starch polymer looked like. Like fat, all carbohydrates contain carbon, oxygen, and hydrogen. Let's review our goal to make sure you met it. After watching this video, you should now be able to use a molecular model to explain how animals grow. If you don't think you can do that, go back and watch the parts of the video you don't understand. Until next time, remember, you can learn anything.